Hello, everybody. Welcome to today's section on taking our peak files from our ChIPseq data and overlapping them with genome features. So we are going to start our official analyses on our custom ChIPseq data sets we made, and we're going to take our consensus peaks from those, or the peaks that overlapped in all the replicates, and overlap them and see how many promoters do they bind, gene bodies, etc. And so really what we're going to do is reapply our overlap skills with some fundamental questions that you would want to ask right away with ChIPseq data. Is where do the peaks land in the genome? Are they binding to promoters, gene bodies, etc. And so we're going to also make a bunch of objects we're going to continue to use, learn how to save our environment and save files so that we can load them faster in the future. So we are starting from here, 05 analyses, and we are doing peak features today and peak features RMD. If you're following along on the website, we are here in 6 ChIPseq. We're officially analyzing all of our ChIPseq data, and that is on this website here, really available, and all the videos are in order of skill sets to learn for that. So let's get to it and get started with the genome features. So here's 01 peak features in our 05 R analyses, peak features RMD, and then I'm going to be doing it from here in RStudio. Okay, so let's get started by just loading in our util function, because we're going to use our import peaks and create consensus peaks, and start to use them as objects in our environment. Okay, so we've got a bunch of peak files in here, and you've obviously cloned this file in the class, so there's some new functions that are in here that I'm just going to use and explain what they're doing, and you can look more into them later. Okay, I'm just going to set my base path to where this folder is cloned, and then the file path to my consensus peaks that we made in the previous class. So I saved them in 00 consensus peaks, and in my 0500, the first class in that we did last. Okay, so here we are in 01 peak features, and now I'm going to run these and then merge this together with file.path, so I can provide just that to list files, where we are going to go to where our consensus path to the consensus peaks we made, and then we're going to look for the pattern.bed, and full names are true, so we can run that. And what we get there is the file path to each of our consensus peaks. I have 7. Depending on how many DNA binding proteins you selected, you'll have a different number. Okay, so we now have a list of where those files are, and we can do a for loop to do import is eventually what we want to do, and we can use lapply as a built-in for loop to go over each one of these lines, and for each one of them in consensus peak files, it's going to do rtrack layer import and give us a list of G ranges of those peaks. Okay. And there we go. We can see our consensus peaks here. They're unnamed, so we're going to want to put the names in there. And the way we're going to do that is if we look at what consensus peaks is, or sorry, in consensus peak files here is what we're going to use. We look at that, it's the file path here. And so to get the gene name, we're actually just going to delete a bunch of the file path and grab the gene name that's in here, like H3K4ME3. So to do that, we're going to do G sub. Here is the first thing we're going to do is take off all of this from the file path. So all of this here. And then we have an OR sign here, which is the pipe means take off this or this string, and that'll take off this .consensus bed, leaving us just with the name of the DBP there. So it's a little bit different than the string extract, a little bit simpler if you want, but it's just a good way to know what is the data that I have that I can grab the gene name from. And we're going to do that here. And then we're going to make the names of each consensus peaks entry the name of that DNA binding protein. So we're going to name it basically. Okay. And the G sub was substituted with nothing is what that is. So it wants to know what is the string you want to substitute, what do you want to substitute it with, and what data do you want to do this on? Okay, so now if we look at our consensus peaks, oh no, okay, file path error again. Sorry, I have these in the folder consensus peaks, so I had to add that. And I'm substituting that off the file path name, substituting with nothing in this object. Okay, so now we are going to load in our GenCode GTF that we did previously in our basics. So if you haven't loaded this, there's information in several classes of how to get it. I'm assuming you just have it by now. So what we're going to do is load in all the features of the genome, all the exons, the introns, and we're going to just focus today on getting genes, which are comprised of a few categories, but we'll focus on link RNAs or long non-coding RNAs and mRNAs. And so I've already done that. That takes about 5 minutes to run. And we're going to, again, save at the end of this so that we don't have to do these longer processes anymore. Okay, so now that we have an object GenCode GR, which we've explored before, where there's a lot of information in the element data, list factor of gene type, gene name, etc. And we'll start looking at those as we go along. Okay, so the first thing we're going to do is some indexing here. So again, square brackets means we're going to take the previous object and subset out another piece of data and call it GenCode genes. And so GenCode GR has all the annotations. And so there's GenCode GR type, which we can look at here, which is a bit messy. It has all the entries, but we can just table it as well and see what the entries are. So in GenCode GR type, what we can see is that there's gene that has 60,609 entries, and that's what we're going to index into with this equals sign. So this is the one we want. You can try other ones, and there's an exercise at the end that involves trying to find overlaps with a different feature. And so you can just subset into here, say in the type, I want only the entries that are gene. And so if we do that, run that, what we can see is GenCode genes is 60,609 long. And so it's our new subsetted G range of features of the genome that are just genes. So it will still have exons in it and all the other information associated with these genes, but it's just now filtered out coding sequences, start codon, and other aspects. Okay. So now we can export this to our results folder and save it so that next time we could just load it in, do our track layer import and put this file path in, and it'll be load much faster than having to wait that 5 minutes each time. I've already done that, so I'm not going to do the exports here. But really what you're just doing is we're saying we just made this object GenCode genes, which is the 60,000 genes in the genome, so to speak, and we're saving it to this file path of the 2 parameters. What is the data and where to save it? Okay, and now we're going to do the similar type of thing, but add a little more to it, a little more tidyverse filtering, where now we're going to go into GenCode genes. So we're still doing the same square bracket indexing. So we subsetted GenCode GR into GenCode genes, and now we're going to go into GenCode genes and subset it to just mRNA genes, which are titled protein coding in the GenCode annotation. And so again, you would want to do something like that table we just did and see what is in there. What are the entries are there? And in this gene type here, we're going to look for protein coding. which is here and has 19,000 entries. We're also going to be looking for link RNAs, which are up here and have 16,849 entries. Okay, so that's what is in GenCode genes gene type. And then, so now we are looking at this information and we're saying what entries match protein coding. And so that is all mRNA genes is now going to contain is the subset of GenCode GR, that's GenCode genes. And now we're subsetting out just mRNA genes. Okay, cool. So that's just this one line here. That's what we are running now. And subsetting, this is what we're going to do this a few times just to get the process down, but this is just a great way to make objects that are subsets of other objects. Okay, so you can export that if you want. And then we already ran this table to see what we did. And now if we did mRNA genes here, we would see that we only have the annotation gene type protein coding in there. And we'll look at that in a second as a data frame. It's a bit easier. Okay, so we've made our mRNA genes object. So now we can have an object to take our peak files and find overlaps with all mRNA genes and compare that to long non-coding RNA genes. So we're really going to do the same thing for long non-coding RNAs to sort of learn this through a little bit of repetition, that we have the same indexing going on here. So we can go and we looked at GenCode gene type and we saw long non-coding RNA here. And so we're now going to say, give me only the entries in there that are long non-coding RNA. And voila, we should see about 16,000, which we do, which is the same number of link RNAs that there are annotated. Cool. So now you can export that if you want and then reload it whenever you need it. But we're making objects that we can use over and over again until you want to update your genome annotation. Okay, so we can do both mRNA and link RNA using the same indexing strategy. We're going into all those things that are genes and now taking GenCode genes and gene type that are using the concatenate protein coding or link RNA. So we did that individually with one object in there, either protein coding or link RNA. And here we're concatenating it so that we can just grab both at the same time. So let's make that object, go ahead and export it. And so in the future, you could start with this object, mRNA and link RNAs, and do that sub-indexing, our track layer, import this, and use the square bracket indexing to separate out link RNA and mRNAs after that. Okay, so we've made objects that now we can just load in and move forward with. So let's try one here. I hope I've written this one out since I didn't do a few of them. But so let's say we wanted to just start fresh. We can just read in link RNA and mRNA genes, and that's how fast it goes. We don't have to do all that subsetting again. Okay, so we can also look at this as a data frame. Right now, our link RNA and mRNA genes is Granges, where we have seq names for the chromosomes, ranges, the starts and widths of features, etc. But it's kind of difficult to look at it that way. So we can just turn it to a data frame by taking it through the tidyverse pipe and using as.dataframe function in R. Okay, so now we have it named now as DF for data frame there. And need to expand this here so we can go now look at this. And we have the start stop with the gene ID. The gene type here should only be link RNA and protein coding. And that's what we see because that's what we sub-selected for. So this is what was kind of going on behind the scenes of that indexing and what we got out in the end. In a total of 36,814 entries. Okay, awesome. So we are getting set up. We have all of our sort of basic gene annotations for long non-coding RNAs and mRNA. What is a gene is a complicated question. So we're just simplifying it with these two categories and showing you how you can pick other categories that we saw go by. Okay, so now what we're going to do is make promoters. So Granges has an awesome function called promoters, and it makes promoters. So basically we'll take the start of a gene and you feed it how much upstream and downstream sequence you want in your promoter. Take a look at it here in the manual. Again, I'm trying to go through this just to get out the concepts as quickly as possible. So pause and go over things as you need. Okay, so we're going to do promoters on an object. So we need a Granges to do this, and it's going to go in and look in the TS or the start site and add 1,000 base pairs upstream and 1,000 base pairs downstream. And so we're going to do this for lincRNA, mRNA promoters. So we're going to do this for all genes, or all of the genes we're looking at here. So we run it, and voila, we have lincRNA and mRNA promoters is a Granges with 36,814. And if we look at the ranges, they should all be 2,000 because that is the size we selected upstream and downstream. And so we can also check that with a width function. And we see that we have all of our entries are 2,000 long. And we can now export that as a feature that we can just read back in the future if you just want promoters back. Let's store it in our results. Okay, so since we have data frame, it's always handy to have a bigger data frame and subset from it. And so, but you don't want a data frame too big with too many things that it's a mess to subset from. So there's a balance to be sort of had here. And so what we're going to do is sort of have a way in the future, whenever we want long non-coding RNA genes, we're just going to make an object to index in with. And so that's going to be lincRNA gene IDs, where we're going to take mRNA and lincRNA's column gene ID and subset it to only the gene type long non-coding RNA. And so if we want to look at this, we can see what this object is with a table and see what we're looking at here. So it's the number of entries for each gene ID here, and then we're gonna subset it to only those that are long non-coding RNA. Oh, sorry, in here is what we want a table. There is what we wanted. Sorry. So we're indexing into the information we want is only the things that are long non-coding RNA in here. And that's why we have the equal sign. And we're doing that in mRNA lincRNA's data frame in the gene ID column and only subsetting to these ones here. Okay. So it's good to check, what am I looking at, where am I in my data frame, and what am I subsetting? But we're really just using square brackets and running a thing to say it has to equal this to subset the data. So we can run that. And we can do the same for mRNA by going into this gene type here and selecting protein coding instead there. Okay, so we'll run that. Excellent. So now what we can do is start to make a data frame and start to collect all this information about the number of promoters, lincRNAs, mRNAs, overlaps, and et cetera. So we usually just start small, build a data frame, and then add columns to it. So here we're using the data frame function to set up a data frame, naming the column here, DBP, as the names of the consensus peaks, which we know is our DNA binding proteins, of interest, which are here. So we have a row, the rows will be named the things as are each of our DNA binding proteins in column 1. And then we'll have a number of peaks where we've done this before, where this S-apply or for loop over consensus peaks will tell you the length of each entry and put a column in there called num peaks. Okay, so let's read that and start to go with that num peaks df. Okay, excellent. We see that we have this protein has this many peaks, etc. And then now we can just keep building to that, where we can add another column by just indexing into a column we want to make here with the cache sign, and then the column we're going to make. And we can do sapply over consensus peak. So for each of those G ranges, run function X, which is now X is imported as what's in consensus peaks, sum the width of X. So basically it's going to be the sum of all the peak widths in one go. We just did length to find how many peaks there were, and now we're going to sum the widths of the peaks and see the total genome coverage that the DNA binding has. And so now if we look at num_peaks_df, we have a new column here with the total things. We could make this in millions and make it a little easier to read. Okay. Now what we're going to do is start counting features and doing some overlaps. We've collected, excuse me, we've collected some features about our chip data and are starting to move forward and analyze it. And now we're going to see how these peaks overlap features in the genome. Okay, so we're going to do that by adding in a new function time. It's in your util my class functions, and it's counts peaks per feature. Okay, so this is a handy function. You can look at it. We've done enough with the create consensus function, but it's kind of a similar idea. And what we do is we provide it G ranges of annotation. So in this case, like link RNA and mRNA promoters here. And so we can click on it here as well to look. What the parameters are is what are your features and then your peak list. So we can do features and then peak list for our given DNA binding protein. And it has two types, counts. So it'll do count overlaps and tell you how many times that feature had an overlap from your DNA binding protein. And then we also have occurrence, which gives just a 1 or a 0 if it did overlap. And this will be really handy for finding similarities between samples, and then running that on 480 samples later will be super handy, and will give us a big matrix of every promoter or every feature and whether there is an overlap and then how many there are as a separate object. Okay, so that's counts peaks per feature. You can take a look deeper. And so we'll first run it with counts here over link RNA and mRNA promoters. And we can see we get promoter peak counts here, which is a vector across all the length of it. The number of columns is 36,000. Each one is a promoter, and the number of rows is 7. And I'm not going to open that because it's huge. Okay, so but we can do row sums for each of these 7 DNA binding proteins and see how many total overlaps there were by using row sums. So we've just made promoter peak counts from this count peaks per feature. And if we sum the row, a row is a DNA binding protein, and the columns are a promoter with the number of counts in them. So we're going to now add this to our num_peaks_df, just doing some index playing around here by indexing with a cache sign to make a new column called peaks overlapping promoters, and do run row sums for each of those rows. Okay, cool. So now num_peaks_df has peaks overlapping promoters, and we can see here now that 4,000 out of 7,000 overlap promoters are a vast majority, whereas very few for this gene there. So we're already starting to learn that there's differences in whether our DNA binding protein likes to bind near promoters or not. And we did all that with that handy count peaks per feature function, which we can keep using in class, where each row will be your DNA binding protein and the columns will be promoters, and a matrix of 1s and 0s, whether there's an overlap or the number of counts. Okay, so now we can split that into the number of overlaps of promoters into lincRNAs and mRNAs really quickly with our handy lincRNA mRNA gene IDs that we made. Here is a value. And so it has all the values for lincRNA and all the values for mRNA, and it can separate those out. So we're doing a subsetting again, using on this side a little extra indexing than needed, but making a new column called peaks overlapping long non-coding RNA promoters, and doing the same row sums over promoter peak counts, but now just subsetting the columns that match lincRNA gene IDs. So this will just row sum only the promoters that were lincRNAs here, and we're adding it now to numpeaks df by indexing into a new column we're making. Okay, so now we can see with numpeaks df, we have of those 4,000 promoters, only 1,000 overlapped lincRNAs, and there's about twice the equal numbers of lincRNAs and mRNAs. So that shows that you'd expect 2,000 by chance, and it's a little lower than that. And so we can investigate that a little bit later too. Okay, so we do the same thing for mRNA gene IDs. So now it's just going to go into that matrix and grab the columns that match. Rows are going to stay the same, our 7 DNA binding proteins in my case, and the columns it's going to subset out are these mRNA gene IDs, and then do row sums across that. Okay, and so now our numpeaks df has some pretty cool information in it. Tells us number of total peaks, total width they have, peaks overlapping promoters, long non-coding RNAs and mRNAs now separated out. And so we can start to see some differences and things that seem to have a lot of peaks on lincRNAs versus mRNAs. And we're going to just continue to explore what happened there. What are the different properties of your DNA binding proteins? Okay, so we can write that out with write CSV, numpeaks df, as we just looked at, and the file path. Okay, so for gene bodies, we can do the same thing, where we can run count peaks per features to find out. Now we're just adding a new feature to this function, so it's pretty malleable, just need a feature, and it'll tell you the number of times your peaks overlap that. So we're gonna do that over mRNA and lincRNA genes and do counts as we did before. Okay, so now what we're doing is making a new column in numpeaks df, counts overlapping gene body. Again, same trick, row sums over the gene body peak counts. And so we're basically just saying take the rows, the total number of overlaps of lincRNA and mRNA genes. Okay, and now we do the same thing for lincRNA genes, where we're taking all the rows for the 7 DNA binding proteins, but only the columns that had counts in lincRNA gene IDs, and then we'll do the same for mRNAs. Okay, so feel free to pause this and go back. I'm trying to do it through a little bit of iteration here, doing the same thing over and over again with indexing strategies. So take some time and pause it and go back and think about it too if you need to. So we're going to write that out. Okay. And so the last sort of step we're going to do here is add some other features to our numpeaks df, and we're going to continue to grow this numpeaks df across the class and add a bunch more columns to it. And so one thing we might want to add to it is what type of DNA binding protein is it? What type of domain does it have? Is it a transcription factor or not? And so there's a paper in Cell that has that sort of annotation. And here's some code how you can download in just R itself. And we're going to make an object destination URL in my results folder here and call it this name of this Excel file. And then we can do download file function, the URL, which I need to make as well. And then the destination for the URL. And voila, I am downloading the file. I've downloaded it. And it's in my results folder here as TF annotations XLS. And we can view the file and see what we got. But let's load it in first into R. And we can do that with read Excel. which is a function that can handle Excel code. And so it's going to say that it's a sheet of 2 page and go to page 2 and skip line 1. I don't recommend learning Excel functions in R, but you could read it in on your computer in Excel. Okay, so we're going to read that in and call it human TFs as an object here, and we can look at it and we see a bunch of columns here, like things that we might want as the DNA binding domain, what type of the DNA binding domain it is, and features like that. So there's column 4 was tricky, so I had to go back in the paper, but that's the one that indicates, is it a TF? And we're going to change the name of that to is TF through just indexing into the names of human TFs in column 4 of that Excel sheet and rename it as is TF. So now when we look at it, we can see this column is a little more informative than just the number 4. Okay, so now what we can do is find the names in human TFs. Here we have these names and gene IDs to intersect in with our DNA binding proteins that we have here. And so if we look in our NumPeaks DF, we have a DBP column here with the name of the gene. Okay, so then we can go in and filter the names in that file we just loaded down in the name column that matches the DBP column in human TFs here. So we're going to match these name with the DBP in NumPeaks DF here. Okay, so we can run that. And the to lower is just making it to lowercase, just in case people spell things different. And we can see that 4 of my 7 have, since we ran length on that filtering, 4 of my 7 have entries in here. And so I'm going to add that human TFs and just grab the first 4 columns of that information. And so I only want in human TFs, the ID, the name, the DBP, and whether it is a TF for now. There's much more information in this file that you can look at and see if you want to grab. But for this exercise, I'm just going to grab the first 4 columns. And so we're going into human TFs, subsetting in with square brackets again. We're doing a lot in these square brackets, but we're basically doing that same filters. We're going into the name column in human TFs and saying it has to be in my NumPeaks DF DBP for the DNA binding proteins I'm interested in, and then return the first 4 columns. So these will be the rows as this subsetting, and this will be the columns. Okay, so let's look at that now and look at our human TFs is shrunk to 4. And these are the 4 proteins where there's information, and only one of them is a transcription factor category. So depends on what you picked. And when we do the large data set, you'll get a whole compilation of different types in here. Okay, so now I'm going to rename the columns there in human TFs to what I would prefer to have them called as ensemble ID instead of ID, etc., DBP instead of name, DBD, and TF column. So I'm just renaming them by saying enter into the names of that object and change the columns to those names that I'm putting in this order. Okay, so we did that. And now our human TFs has our new ensemble ID and has a bit more clear information in it. Okay, so now we are going to merge human TFs with NumPeaks DF, and we'll do that just with the merge function in R. We did a left join before. We could do the same thing, but we're going to do merge and put it into NumPeaks DF and rename it by putting NumPeaks DF in and human TFs to merge them, and all the examples is true. Okay, so now if we look at NumPeaks DF, we see I still have all 7 proteins here, but now I have this information and NAs where these proteins just didn't have entries in that file. Okay, so numpeaks DF is getting bigger. We are almost done there now. And we are going to just look at the dimensions of that. That is NA. I have 3 of them. And so that's fine, that's what we're going to do. And then I can take here and just take the first 12 columns from numpeaks DF and rename it here. And now look at numpeaks DF. And we have all of their information, including our whether it's a transcription factor or not. Okay, so the last thing we want to do is run an occurrence matrix, which is the same thing we just did. So I'm going to go over this very briefly, where we use count peaks per feature, but we're changing the type to occurrence. And so this is a good exercise for you to go forward with and just run it on the objects we did the same up above with counts. But just looking at the indexing here, we're running counts peaks per feature, and the parameters are the features, and the peaks are your consensus peaks, and the occurrence just means it's going to put a 1 or a 0 if it overlaps. And as a refresher, all the columns are promoters for link RNAs and mRNAs, and the rows are your DNA binding proteins. And so we can make sure this is all set to run and looks good, and make sure that ran. So this is just a double check to make sure the columns are in the right order. And now you can write it out. And then you can do that by also subsetting the gene type. So we can make the same way we made a numpeaks DF. We're making a data frame here for the peak occurrence data, where the gene ID is the call names in promoter peak occurrence, and gene name is in the gene type column. So we're just doing some indexing into these objects, which we've done up above, and adding that information as column names here. So we can just run this whole data frame construction there. We also did some call sums there to, the number of DNA binding proteins that bind to a given promoter was defined by call sums of peak occurrence. Okay, so we can write that out. And the last thing I want to show you is that we've done a lot of work here and made a lot of important things so that we would want to use in the future. So we're going to do the coolest thing is save it all. And so we can just load our environment right back up from empty. And we just do that with simply the save command here. And then all the objects in your environment that you want to save. And so we want consensus peak, gen code genes. We want all the stuff we made. And in our next session, we can just load all this in and go forward. And that's what we're going to do. So let's do this exercise. It's really nice how this works. And so we're going to save all of these things, our peak occurrence data frame, numpeaks DF, all of this we're going to use in the future. And we can just hit save. It was that fast. And it needs to know the file path is the last parameter. And I just saved it to my results peak features. And you can see it was just saved now. And it's called .r data. And so now I can just erase my environment here, which is kind of scary when we've done so much work. And then now I can just use load and load that in. I have the command verbose equals true because I like to see what objects come up as it's loading. And so all we have to do is run that, and voila, our environment is back and we're ready to do more overlaps and more plotting, etc. as we move forward. Okay, so there's an exercise here for you to find other features to find counts over and add to your numpeaks DF. But we now have a great data frame with a bunch of columns of information, peaks overlapping link RNAs, mRNAs, and we can use this to make plots and other things which we're going to do moving forward. So for now, that's that. You have your consensus peaks for each DNA binding protein and and a bunch of genomic feature objects, and we can continue to work with these and find out more. All right, with that, thank you and be well.